on each of these chromosomes. But each of these colored areas represents a distinct chromosome. I mean, you might be used to thinking about chromosomes like that. Of course, those don't exist. This is a completely compacted chromosome that exists only during mitosis or meiosis, right? In the interphase, remember the stages of the cell cycle in the interphase, the chromosomes are not this tightly condensed. But what people weren't really completely appreciating, and by people, scientists weren't completely appreciating, is just what the level, the three-dimensional organization of the nuclear genome is. So this is just the nucleus. We don't see the cytoplasm. And it's actually been known for probably 15 or 20 years that chromosomes aren't just kind of randomly intermingling with other chromosomes, that there is some structure to this. But there's a lot of research still going on now and will continue to go on just how dynamic but important this overall three-dimensional or spatial organization is to gene expression and controlling gene expression. So each of these, and we'll see this in more detail in a few minutes, each of these is considered to be a chromosome territory. That's the word that's the formal term for it. It's, you know, you might be thinking chromosome read, but chromosome territory. So this is essentially the order that I'm going to go through. So this is the G banding, you know, where you just stain. Now remember, again, these are metaphase chromosomes at the point that they're most tightly compacted. So you have to have cells that are in culture. What they do is, these cells would normally be dividing, but what they do is treat the culture of cultured cells with a chemical that inhibits, that arrests them in metaphase of mitosis. Okay, and then they just do a spread of the chromosomes, stain them with this dye. This is just one of them, Hemsa, G-banding. There's other dyes. And then, of course, the chromosomes aren't nicely lined up like this in pairs. But then back in the old days, they take a photograph of that through the microscope and then print it out and literally cut it apart and align them. Now this is all done very easily digitally. The other thing I wanted to remind you of that you should appreciate, remember these, that this is each a homologous pair of chromosomes, right? And we can identify each of them on the basis of both the size, the position of the centromere, we'll see that in a few minutes, also the pattern of the bands, okay? And this kind of karyotyping is still being done to this day. There are more sophisticated methods, especially involving fluorescence, fluorescence, in situ hybridization fish that you might be aware of. We'll see a couple examples of it. So it's still important, though, to be able to understand this concept of the chromosomes and the fact that the homologs, the homologous pair, are identical to each other with respect, for the most part, which should be with genes. Now remember, there are different alleles on each of the homologs, okay? The other thing I want to remind you about, let's say this homologous pair of chromosomes 4. How many molecules of DNA are actually represented in this pair, homologous pair of chromosomes 4? Molecules of DNA. And just to give you a hint, by a molecule of DNA, it means double-strand DNA. If you want to think about it a different way, how many chromatids are present? Two chromatids? Remember the difference between chromatid and chromosome? We've got two chromosomes here. Who said that? There's 4. Remember, this is at metaphase. Each of the chromosomes this is after S phase, right? Remember G1, S, G2, then mitosis. These are chromosomes in metaphase of mitosis. So each of these chromosomes consists of a pair of sister chromatids. That's what I want, we're not going to be going through any papers in detail about this, but I just wanted to remind you. We see these structures, right, and you think, oh, well, there are only 2 chromosomes there, but remember, each of the chromosomes is a pair of sister chromatids. Okay, so this is what you should remember, a review from Gene 3200. Humans have either, their chromosomes are either metacentric, where the centromeres are about in the middle and the 2 arms are about equal size, submetacentric, where the centromere is just positioned slightly off-center, or acrocentric, where it's real close to the end. Now, I can't remember if this was covered in Gene 3200, but with these submetacentric and acrocentric, you can refer to a short arm, the P arm, or the long arm, the Q arm. If you're wondering what's located on the short arm of the acrocentric chromosomes, there's only, I think there's 3 of them. That's actually these very short arms on chromosome 13, 14, and one other chromosome. These are actually repeats of the genes that encode ribosomal RNA, so the rDNA genes make up the bulk of chromosome 21, of course, yes. They make up the bulk of the short arms. You might be interested in that. Is there a 4th category of chromosome? Telocentric. There are no telocentric chromosomes in humans, just an evolutionary quirk. Now, of course, you don't have to learn all these bands, and you don't have to identify or learn that chromosome 1 is a metacentric, chromosome 2 is a submetacentric. I just want you to kind of know that each chromosome is a characteristic size and a characteristic positioning with respect to where the centromeres, as well as a characteristic binding pattern. Now, I just want you to, this is all. Okay, so I do want you to understand this because this nomenclature is still being used, even when people are referring to genome sequence. If you look in a genome browser online that has literally the base sequence of a chromosome, it will be referenced to that position on the chromosome. So it's still useful to know this nomenclature. So here I'm showing this is just chromosome 5, okay? Submetacentric, centromere. Now each of the arms, there's the P arm, the short arm, and the Q arm. So since this is chromosome 5, it starts with 5P and 5Q. Then the numbering of the bands. So these are the old-fashioned cytogenetic bands that were identified probably in the 50s or 60s, okay? So they number going from the centromere distally to the telomere. Remember, these are linear chromosomes, right? There's a telomere on each end. But for the short arm going from the centromere to the telomere, it moves from 5P12. Now this is not 5P12. It's 1, 2. I don't know why, but and 5P13.2, 14.1. Okay, so moving centromere distally, same thing with the long arm centromere distally. 5Q12.1, 5Q12.3, and so on. Okay. Now I do want you to be able to interpret that, okay? Because what I want you to learn, and this is still being used, this nomenclature is being used in research papers now. When we're referring to the karyotypes, right? Aneuploidy is a big deal in human genetics. We'll talk about that in next week. We won't have any research papers on it, but in case you're wondering, not all research journals make the authors follow this exact nomenclature, but it still is commonly used. There are some journals that insist that the authors do that. So what you have to do if you're following the standard nomenclature is first you state the chromosome number, so 46 for the normal, right, 23 pairs. The sex chromosome constitution, you have to designate XY or XX, and then if there's any kind of chromosome rearrangement, okay, or aneuploidy. So these are the normal, remember the terms euploid and aneuploid, okay? So the aneuploid, 47, that means there's one extra chromosome. This is a female, and she's got an extra chromosome 21. It's not telling you that there's 3 chromosomes 21, of course, but since you know that there's 47 chromosomes total, that means that there's, the normal 2 copies of chromosome 21 plus an extra one. Same thing with trisomy 18, Turner syndrome. You often see it in textbooks, and it's referred to as being XO, but there's one less chromosome, or Klinefelter's, there's one extra chromosome. Gets a little bit more complicated, but you need to be able to refer to chromosome rearrangements, deletions, inversions, and then on the next slide I have translocations. Okay. You still have the chromosome number. Of course, these are all balanced, right? There's no change in the number of chromosomes, but you're either losing or you could gain. It'd be the same thing with the duplication. You're deleting on chromosome 4, so that's the number after the type of rearrangement, chromosome 4, the deletion is within P15. Okay. Same thing with an inversion, a little bit more complicated. Of course, inversions only involve one chromosome. I'm not telling you, well, I've got two examples, para-centric and para-centric. This one involves chromosome 2, this one involves chromosome 14. The first, so the actual inversion breakpoints, the bands that the inversion breakpoints are on, one is in P, the short arm, one is on the long arm. Okay. Same thing here. Both of these are on the long arm. And remember the difference between para and para-centric. Okay. Now the other thing to know, since you're being told that each of these individuals has the normal, the euploid number of the chromosomes, you know that in addition to this inversion chromosome, there's also a normal chromosome 2, a normal chromosome 14. So this individual is actually heterozygous for an inversion, each of these. Translocations, T stands for translocation. Now in parentheses, and now pay attention to the semicolon is important. That's the standard cytogenetic nomenclature. Okay, it's not a comma, it's not a colon, it's a semicolon. T in parentheses refers to the two chromosomes. This is a balanced translocation, so this is a reciprocal translocation. Remember where part of one chromosome is that DNA is broken, and that part is moved over to another chromosome, but that other chromosome was broken, and that broken part was translocated to the other, that reciprocal translocation. This is telling us that there's a reciprocal translocation involved in chromosome 3 and chromosome 9. The band that's affected in chromosome 3 is P14. The band that's affected with chromosome 9 is Q21. Okay. Now this is unbalanced. So derivative, DER, that indicates, and I don't know the official definition for this, but to my mind, the explanation for this. This refers to a derivative or derived. This is a chromosome that has that derived chromosome 3. That is telling me that the translocation actually involves the centromeric, the centromeric region of chromosome 3. Because that's where you start thinking about whether there's non-disjunction or disjunction taking place. Okay, so this is translocation involving 3 and 9, and again the breakpoints are P14 for chromosome 3 and Q21 for chromosome 9. Okay, so I do want you to know that. That's on the study guide. You might have to translate that. Genome sequence. Now, you don't have to, you won't be tested on anything in the genome sequence. I just want you to be aware of some things. Okay. Some online things, a little bit of history of the Human Genome Project. You may already know about these. You can look at them on your own. You might not know this, but what's called the human reference genome is actually composite. It's not DNA for just one person. And that was done intentionally for privacy issues. It was also as a way of trying to make it representative of more than just one person, right? Because everybody has a slightly different genome sequence, right? But they recognize that, and I think Kai-Jan will tell you a lot more about the individual genome sequences that are available now. There's literally tens of thousands of them, right? But this just tells you about the human reference genome. So every organism that's been sequenced, at least that's been around for a while, human, mouse, C. elegans, Drosophila, it's just one reference genome. It might actually have some differences, right? If you look at an individual sequence and compare that to the reference, there might be some, very likely there would be some sequences that are present in the individual genome that aren't part of the reference genome. There can be different alleles, right? SNPs and microsatellites, all kinds of things that are different. Part of that might be because we refer to the human genome sequence as being completed, right? It's been completed since 2003 officially. It's not literally completed. There are gaps that are still being filled in, and it could be that there's a gap that was sequenced in an individual that just isn't part of the reference genome. Now, as soon as something gets published, new sequence gets published, that is added, it's verified, so that's added to the reference genome. But it's got to be kind of validated, and I think probably Kai-John will tell you more about that later on. Okay, and I just love this. Think about this. This was in 2001. The more we learn, the more there is to explore. I think that's an understatement, okay? I think that you can magnify this by 100-fold. We're learning so much from the genome sequence that I don't think they completely appreciate it. And I think there's probably going to be even more in 10 years, 15 years, okay? Again, don't worry about all this. It's just to give you an idea, to remind you of some things of the genome. If you wanted, so this is just giving you an idea of the difference between what was considered to be the reference genome in 2014 versus 2018. It's not that the genome has expanded in about 300 million by 300 megabases. That's how much has been filled in. But when you think of that being out of 3 billion, right? It's not very big. Some of it is because some regions are just hard to sequence. And there can be some chromosome regions that explain that. And just to give you an idea, now the total number, okay, one thing I didn't point out, or you might not be aware of, remember those karyotypes, the karyograms and stuff like that I showed you previously, and the numbering 1 through 22, and then the sex chromosomes, they're numbered in order, the largest being chromosome 1, and the smallest ones being chromosome 22. So you might think that, kind of the distribution of then the number of genes for each chromosome might be proportional to the size, the amount of DNA on each chromosome, right? You would think that the largest chromosome would have more genes, but that's not the case. And that's just something I want you to appreciate. Actually, the gene that has the most number per megabase of DNA is chromosome 19. I don't know if there is a biological significance to it. It might be just because that's the way it evolved. Okay. It could have some biological significance, but who knows? Okay. Of course, the Y chromosome is by far the smallest and by far has the least number of genes. Not that you guys aren't important. There are some important genes on the Y chromosome, but okay. And this is just, again, I just pulled out some example chromosomes. Here's chromosomes. Now, all of these, this is not relative to size. I've stretched these out so they're all the same size. But even though the gene density varies along the entire chromosome, even when you look along the length of one chromosome, the density within each region varies. It's not randomly distributed equally distributed along each chromosome. It's not even on either of the arms. Now, the difference here with the Y chromosome is unique is that all of the genes on the Y chromosome are really located on the end. There's this entire region of the Y chromosome that is worthless. Now, it's still probably important, but right. And there's some really interesting explanations for it. And evolution of sex chromosomes is actually something very, very interesting. And a few more statistics and things that you can read about. Repetitive DNA, just to give you an idea, you can take a look on your own. Examples of some of the browsers that are available if you wanted to look at. And I think I'll just take a look at the rest of the slides on your own.